So welcome everyone to the CGIL seminar. It's Ivan here, and today we are delighted to have with us Dr. Huson. So to introduce our speaker, Dr. Heather J. Huson is an associate professor of animal genetics in the Department of Animal Science at Cornell University. Dr. Huson received her bachelor in animal science at Cornell University and PhD in molecular genetics at the University of Alaska. A significant part of her PhD work was done at the National Institute of Health and the Human Genome Research Institute, and her postdoc research was with the USDA Bovine Functional Genomics Laboratory. She has a diverse background combining animal breeding, veterinary technician experience, and molecular genetics across livestock, companion animals, and wildlife species. Her research aims at improving animal health and performance by investigating the genetic regulation of economically important traits. In addition, she explores population structure and admixture to better understand selection, breed development, and conservation. She focuses primarily on working dogs and dairy cattle genetics and selection. Dr. Huson manages both graduate and undergraduate research within her lab and teaches courses on animal genetics and applied dairy cattle genetics. She's also the director of the Cornell Raptor program, which promotes education, conservation, propagation, and research. So welcome, Dr. Huson, and I'll pass it over to you.
All right. Thank you guys so much for having me, this opportunity to give a lecture to you. I really thought about, how I could contribute to your group and what this lecture should look like. So while I'm gonna definitely show you some results of my research, I've also included information related to kind of my research design and the reasoning behind my research design. And I put this in here with the idea that I know there's graduate students and postdocs in this group, and I know that's a big picture when you become, new faculty or new researchers on your own to figure out where do you start and how do you do this. So I'm going to kind of show you how I did it. So hence the complexity of the phenotype, population, and approach when we think about genomic research and interpreting that research. So I put this up here, papers all about phenotypes and genetic selection, because it just exemplifies the importance of generating data for use in genomic research. And oftentimes it's a balance of quantity and quality of data that as researchers, we need to figure out how to do this when we have, limited funds, there's logistical concerns, we need to reach statistical power. There's all kinds of things that go into that. So when I started formulating my own couple research projects, when I started out as faculty, I had kind of the same approach that I did for two of my main projects starting forward, and it's worked well for me. So one of the things that I was really interested in doing is creating these kind of longitudinal studies where I could get really extensive data across lactation in cows. And it also allowed me to kind of set this up as a prospective data collection. So I wasn't kind of looking back and saying, what can I do with the data set? I was looking forward going, how do I want to set this up for the best data set I can collect? With that said, I aim to work with commercial farms. So there is a bit of logistical limitation there in convenience sampling that worked with the farm because they're a commercial farm. Then I often went on to the part of, after I collected this data, the first step was not genetics. It was really to do an epidemiological analysis, so really statistics, and look at, how these traits related to the disease that I was interested in. How did the traits, the multiple variables I collect, correlate to one another? And then I want to use that to inform my genetic research. So for me, I was using primarily SNP chip data in the two projects I'm going to be showing you today. Well, three projects actually. And that was the one that I chose for the amount of data I could get for the number of animals I had. So phenotype was a large portion of my study that informed how I went about the genetic analysis. And for me then, the deliverables of these projects we're really identifying cow level risk factors of the disease. So we're not even at the genetics yet. We're just thinking about risk factors to the cow related to the disease of interest. But then moving on, I did do the genetic analysis, and therefore we're identifying markers related to these different variables of risk factor, and also think about how do we include those risk factors into genomic selection. And lastly, what's the biology under these things? What are some of the biological implications of how we're regulating or how genetics are influencing complex traits? And so for this, I'm going to talk about two of the studies that we've done in my lab. Some are still ongoing within this, but really focusing on mastitis and lameness. So for the first one, honestly, my graduate student at the time, and now she is a doctor and full research scientist within the USDA. But this is Dr. Asha Miles. She did the majority of the legwork in the analysis for our mastitis project. So when we started the mastitis, these were my aims. I wanted to start out and start characterizing things such as the udder and teat conformation and how that relates to mastitis. Of course, I wanted to do genome-wide association studies looking at udder and teat conformation, not only in and of itself, but also in relation to mastitis. Then, of course, we have used somatic cell score for decades now in a selection against mastitis, but I wanted to look at this a little bit more in depth and say, is there a time-dependent genetic pattern in the response to somatic cell score across lactation? I also wanted to look at population variation related to those animals that are the most susceptible to mastitis, as well as those that are the most resistant. And lastly, we're looking at milk microbiota profiles and its relationship to mastitis. So at the moment, we've accomplished tasks 1 through 4. We are still working on task 5. And one of the big things that I did in setting up my study design was I was setting up a prospective cohort study design, and it was longitudinal. So from this, we set up basically collecting data from key biological time points. So a lot of times when we do these longitudinal studies, you'll often see that it's like, every day, every week, or every month, or every couple months. I focused on biology. So instead of being consistent on weeks, days, months, I looked at when are the key biological time points for a cow throughout a lactation? When are they going? When are things really changing that might affect the body and how the body responds? So for instance, I collected samples on basically within day 1. So they calved. This is parturition, and we have colostrum production. Then we went on to day 3 to 5. And this is very early lactation, but it's kind of as you're transitioning from colostrum into baseline milk. Now, days 10 to 14, this is usually peak mastitis incidence for a lot of cows, and it's also a significant time of negative energy balance. Then we went out, we extended our time point out to 50 to 60 days, where we're moving into more of a neutral energy balance and an end of the voluntary breeding time primarily. This took us out to 90 to 110 days, and this was really peak production, early pregnancy, the cow's into the swing of things here. But now we finished this off with really right near dry off. So our samples were before they were dried off, but it's getting close to that dry off in later pregnancy. So this is one of kind of the unique things. Not only is it longitudinal, but I focused on biological time points, not just consistent days, weeks, or months. So for those time points, we worked with a commercial farm. We enrolled 523 cows. We followed them across lactation. At all of those time points, we collected milk samples. Upon enrollment, within those first 2 collections, we collected blood samples. At 2 different time points here, at the 2-week time point and then out at 90 to 110 days, we collected conformational scores on the udder and teats, looking kind of, early on, but then also double checking, was there any change later on? And lastly, we brought in data collected from dairy comp from the farm records, as well as our genomic data, plus all the data our researchers collected through this, like the conformation scores. Outcomes, like I said, our first outcome was an epidemiological study. So we're doing statistics on udder and teat conformation. We were able to come back and say, here are the risk factors, cow level risk factors related to clinical mastitis and elevated somatic cell score. It was cell count changed into cell score. These down here, these were just related to clinical mastitis, but these are really informative things for farmers just to look at in managing their cows. You know, we didn't even get into the genetics, but we're producing, you know, some helpful, useful data for farm management. With this said, and I'm not going to show this part to you, we did do genome-wide association studies that were published on this of all of these different conformational traits for udder and teat. You know, when we think about udder, we were looking at the udder attachment. When we were looking at the teat, one of the unique things we looked at is teat shape. So if these different types of teat shape, whether they were flat or rounded or very pointy, that was one of kind of our novel traits we looked at. Some of these, like udder conformation and some of the other teat scores, are traditionally scored within the Holstein Association. So we did, you know, our epidemiological study. We have risk factors associated with mastitis. We were able to put all these risk factors into a single index and look for common genetic variants associated with that, as well as creating, you know, just identifying genetic regions associated with all of these conformational traits. So that was one of our big outcomes, but I'm not going to show you all those Manhattan plots for this one. What I do want to focus on is one of the more thought-provoking points of this study. And one of these was really thinking about somatic cell score and how we use this in selection in dairy cattle. Like I said, we've been using this for quite some time now to select against mastitis. And somatic cell score, everyone knows, is highly associated with mastitis incidence. With that in mind, what we commonly do in selection indexes is really use a cow's median lactation somatic cell score for selection. This is the information we use. So on our specific data set, we ran this, and we have kind of lines of significance for Bonferroni correction. We have the SNPs here that you can see highlighted. These pass false discovery rate, multiple testing correction. So it's not a very exciting Manhattan plot, but it is what the industry uses, and this is on our particular data set. So remember, we only have 525 or so cows in this data set, and a lot of people cringe. If I could see you and we were in person, I know I would see some of you cringing on a GWAS with 500 animals. But here's the kind of the benefits of our data set, is that we were really interested about what's happening across lactation. Is there change, is there variation across lactation in how the body responds to elevated somatic cell count? So here we have all of our different time points. We ran GWAS at each of these time points on the same cows, okay? Within that particular data set, you see some of these, you know, it was basically what are the cows at each time point that had elevated somatic cell? So these were quantitative traits that we were running. But we have, you know, everyone that's in blue, the SNPs that are in blue, these are the ones that pass false discovery rate. The lines are representing Bonferroni. So you do notice not all of our GWAS plots here show SNPs of significance. So let's just focus on the ones that did. So again, sometimes we get kind of, I guess, kind of stuck in a rut of maybe just being a pure geneticist and not really thinking about the biology. So the bottom line is, yes, I can run a GWAS for elevated somatic cell count or score on days, you know, at calving, but a cow should have elevated somatic cells at calving. That is a biological response to calving. This isn't necessarily mastitis. So in reality, going forward, when I think about mastitis, I'm going to focus on these particular time points that gave us results, and they're more getting at actual mastitis incidence and not a normal biological response to something different than mastitis. And lastly, I'm going to pull up that GWAS on the median for comparison. And so the bottom line that we really tried to get across with this is, it is not my expertise on genetic selection, on the algorithms of genetic selection. But looking at these and thinking about how do we select for somatic cell scores in cattle, we're using a median. And it's kind of capturing lots of different stuff that you see in all of these. But I think it's really important for us to consider as an industry, is there a way that we could potentially improve selection against mastitis if we targeted different regions at different time points across lactation? While computationally, this might seem like, yeah, we aren't even going there yet. I think down the road, it is possible, and we could possibly incorporate this and make our selection, improve our selection, and also improve it from a biological standpoint for the cows. So this is kind of one of the big outcomes that maybe it might not be monumental to some people, and they might think median, hey, it works just fine, but I think we can improve it both as an industry and also biologically for the cow's benefit if we consider time points. So that was just one of our outcomes of this study. Another way that we approached this study is we wanted to look at populations, and are there unique genetic patterns related to populations of mastitis cows? Okay, so what the heck am I talking about populations? So what we wanted to do was, using our somatic cell count data at each of these time points, we wanted to look at cows that are always healthy throughout an entire lactation versus cows that kind of swing up into elevated somatic scales, then they kind of cure, they go back and forth, versus cows that are just elevated somatic cell count all the time. So what we did here, and I know these are small, but focus on the colors. I'll describe the rest. So we have all of the different individuals within the plot. These represent our cows in our study. Down here in green, we have cows on the x-axis. This was their somatic cell scores at days 10 to 14. Then on the y-axis, we have somatic cell scores on days 50 to 60. So between these two time points, at these two time points, we have animals in this lower left quadrant that at both times they had low scores, they were healthy. In this upper left-hand quadrant, we have somatic cells where they were originally healthy at the first time point, but then they elevated, so hence they're new cases of subclinical or clinical mastitis. Now I'm going to jump down here into over on the right-hand side. On the right-hand side, we have cattle that at both time points, they had elevated somatic cells. So they're kind of our chronic animals. They had elevated at both time points. And in the lower right-hand side, we have the animals that they were elevated at the first sampling, but they actually got better. So these are our cures, okay? So these are the first two time points when we think about mastitis. Now that's moving, we put the 50 to 60 days on the x-axis, and we brought in a new time point, the 90 to 110 days. So we left the animals at the same color they were, but look how some of these animals have moved quadrants. Some of the healthy animals became new cases. Some of our kind of new cases are now chronic across these two time points. Some of the chronic actually cured by this last time point. And we bring it to the very last time point. You can again see how the animals have moved. And within this last one, it's hard to visualize on here. Take my word for it. We actually identified the animals that, regardless of time point, they stayed in this chronic point every single time. We also identified the animals that stayed in this healthy category every single time. And then lastly, we put these animals that kind of moved back and forth. These were kind of our average animals. So we made these three distinct groups: the healthy all the time, the average that moved around, and the chronic all the time. And most importantly, I was really interested, was there genetic variation in these healthy and these chronic animals? So one way that we decided to look at genetic variation was actually using Wright's F statistic and FST for fixation index. So really we're comparing in this way two populations. So it's kind of like a case control, but looking at allele frequencies. And you have one population over here with your second population over here, and you look at this allele frequency difference that ranges between 0 and 1. So really, when they are completely distinct, you have these factors where they're opposite here. And then you have situations where there's commonality among the two populations, and your values are in between here. So using FST, we looked at that allelic differentiation among populations, and we have average versus healthy. So remember, this y-axis is on a scale of basically 0 to 1. And we put our threshold as we wanted to get FST that was greater than 0.4. We got nothing on average to healthy. It's not interesting. They're pretty much the same. However, when we look at average to chronic, we see that there's variation that's starting to separate these animals. The average animals that are kind of moving back and forth, sometimes they're elevated, sometimes they're reduced, versus those chronic that are always elevated. And then lastly, we put healthy versus chronic. And these are two kind of extreme populations. And again, we have a few places in the genome that are differentiating these populations. And with some of those, we see commonality in comparing these populations. So, of course, we went on to kind of explore what are candidate genes in this region, and how does this relate to mastitis and potentially animals that are resistant versus animals that are the most susceptible. And I think some of the important parts here are even our chronic animals, even though they had elevated somatic cell score the entire time of their lactation, at least when we were sampling, they were also still in the herd and producing. And that's important because these were good producing animals that the farmer chose to keep. In the end, with our project looking at mastitis, I think we did genome-wide association analysis on udder and teat. We did it on somatic cell. We looked at somatic cell across time. We also did marker-based FST, and we looked at variation among populations of susceptible versus resistant. And the bottom line is all of these regions that were identified in our genome analysis really showed us multiple different pathways that are all related to the immune system. And it just exemplifies, but I think it's important to see how all of these things came together. So this is a figure we snagged that shows a really good example of kind of the different pathways of the immune system and how you can work through here. And over here, I'm putting out in our data, we came up with pathways related to toll-like receptor signaling, B-cell maturation, T-cell antigen presentation, and complement pathways. And these stars that are over here, that's where our results led us to these pathways. So I think the important part here is just thinking about the redundancy of the immune system and how essential that is to maintaining a robust host of defense mechanisms. If one thing fails, another thing can kind of capture it and take care of that disease. So that was kind of, big picture. We had lots of little parts in this study, but bringing it all together. All right. So we ended up with 4 papers that will tell you lots of different results and more details about that I'm also happy to explain. But with that, I think the big results for that was we did identify cow level risk factors. We identified QTL related to conformational traits as well as QTL that were time dependent for somatic cell score. We also looked at variation, genetic variation in those distinct populations of resistant and susceptible mastitis groups. And in all of this gave us new insight to the complexity of genetic regulation and response to mastitis. I think some of the key points when I talk about research approach and methodology. So one of the big things here was that our project on mastitis focused on generating more comprehensive data that was longitudinal data, and it focused on biological time points. Our drawback was we didn't have a ton of numbers. We had just over 500 cows, so that was a limitation of our research. But it does give us some evidence that we might think about incorporating in how we do future selection models, whether we want to incorporate udder and teat conformation traits or thinking about time relationship to somatic cell count. So that wraps up all the good stuff I'm going to tell you about mastitis and takes us into what did we learn about lameness. So this particular study was run by my graduate student, who's now Dr. Cassandra Stambuk. She's working on dairy extension at this time point. And she worked on characterizing digital cushion thickness in dairy cattle as a way to tackle lameness. So our objectives in this study was, again, to characterize digital cushion thickness across lactation. How does it change and how is it related to lameness? Then to look at, what are quantitative loci for this trait in cattle? And how does this vary across breeds and sexes? So why did I look at digital cushion thickness? Well, quite honestly, it gave me an alternative way to tackle lameness. So some of the limitations here were there were already studies that I knew that were going on, and some of these are here in Canada with Guelph researchers that are amazing, looking at different specific diseases related to lameness. And I could not compare in a research design with the data they were already collecting. So I looked for an alternative way that I could tackle lameness. And one of those ways was to look at this idea of digital cushion thickness. So it'd be more of an indirect way of looking at lameness. So digital cushion thickness, where is that? It is this fatty pad in orange that is in the hoof of the cattle. And here where this kind of pink spot is, this is really the main area where sole ulcers are commonly found. And what we did was we used the ultrasound to take basically measures of this fat pad in cattle going across lactation. So previous research that was already published had associated digital cushion thickness with claw horn disruption lesions. So hence our motivation to tackle lameness. They also estimated that it had a heritability of 0.33. Fantastic. We can make an impact on this in genetic selection. They had established a routine of using the ultrasound to measure this. However, we were going to expand on that. So we looked at lameness kind of in a couple ways. One, we did collect data, particularly focused on claw horn disruption lesions that were from the farm on sole ulcers and white line disease, as well as any other diseases that occurred, but we specifically focused on these. My graduate student at the time also worked and studied visual locomotion. So she scored the cattle at different time points on lameness based on visual locomotion. We had two phases in this study. The first phase was a very small cohort on one farm, and we wanted to kind of look across multiple time points. So we had 4 time points, and this logistically was really tough because we were trying to not only use previously published data to say what are the best time points to collect data, but also to think about what are the best time points that work for the farm. So we were trying to coordinate this with hoof trimming. So bottom line, we had one time point while they're dry, that's roughly 40 days prepartum. The next time point was after they had calved, so within that first 30 days of calving. And then we went on to later in lactation, 90 to 100 days. And lastly, we looked at near the very end of lactation, basically into dry off again. So these are the time points that we collected ultrasound measures. We also have our blood sample for genetics, plus we had things such as the body condition score, locomotion, lesions that had occurred, as well as animal height. So these are kind of all the different measures we pulled this on. And the graph up here is from the previously published data and their findings of digital cushion thickness and its relation to claw horn disruption lesions. So we were focusing on when it was really thick, when it was really thin, and when they didn't have data. So that phase 1, small cohort, multiple time points, what did it tell us? It told us that digital cushion thickness did vary by time point. It told us that it was thinnest when they were dry before calving, as well as for premiparous animals. It was also thin here, kind of in mid-lactation. But it was always thickest, regardless of premi or multiparous animals, here at the end of lactation. Another thing we were able to do then was for our research purposes, and that was because we needed to identify the prime time points of thin and thick to sample additional animals. So we accomplished that. Next thing we identified is that digital cushion thickness, it does vary by digit. Now our study focused on the right feet, both front and hind, where the previous studies had only done one of the feet. So we looked at right feet, front and hind, and both digits on the right feet. So here we see the blue one is showing us these were consistently our thinnest ones, where we had thick ones on both a front and a hind foot. The other things we were able to determine is that out of all of these digits and the changes over lactation, if we use the hind lateral foot at time point 1, so that, very first one here, this was a good predictor, significant predictor of claw horn disruption lesions that would occur later in lactation. The other thing we could use was that same hind lateral as well as the front lateral at time point 2 could be used as predictors for locomotive lameness later in lactation. So our goal here was really to look at early time points and see, could we predict lameness later in lactation? This took us then to our second phase of this project, where the bottom line is we needed more animals. So we need more animals, but because we were having more animals in multiple farms now, we went to 5 farms, we really had to kind of reduce our sampling. So by reducing that sampling, we focused on just 2 time points. So where it was consistently thin and where it was consistently thick. So this is what we did here. And at those time points, again, we got body condition score, we got height, we collected our blood, and we were doing ultrasounds on the animals' hooves. So with this in mind, what were our conclusions? What did we figure out from this? We were able to, again, validate, but in a larger cohort across farms, that digital cushion did change over time. So, but it was consistent. Thinnest time was still the thinnest time versus thickest time. We also were able to validate our initial patterns of what was the thickest and thinnest digits. Now here, this did change in that one front or hind digit was not showing up, but this was still the thickest and this was still the thinnest. And lastly, we were able to establish with this larger number of cows that digital cushion thickness also was correlated to body condition score. So the animals that had a lower body condition score, as you would expect, also had thinner digital cushion thickness. So less fat preserves in this area. So on to our genetic study. So I will say, I had a couple people come when I set this up and they said, well, you're fishing for p-values. And I don't like that. I don't like scientists that do that. And I took it very personally. But I said, I'm not fishing for p-values. I have different questions. And I think that's really important because we did, and trust me, if my grad student, if I would have said do 1 GWAS, she would have been so happy. Instead, she did a lot. But the bottom line is we were asking different questions because I wanted to understand the biology of this. So we had all of this data, let's use it. So we were able to look at association studies to say, was there genetic variation related to the digit? So how much thickness was related to the digit? How much thickness was related to a foot versus time point? And now when we did all of these, I will say, I'm not putting the data here and it's just too much, but we did account for when their hooves were trimmed, who the hoof trimmer was, what stage in lactation that was all done. So we also had these covariates that can definitely affect our measurements. And in the end, when we wanted to know what the heck, how does genetics affect and associate to these things, we found that yes, there is a correlation. We could find genetic patterns related to the digits per sampling time point. So there was a relationship between digit thickness and time point. And that came out in the previous work. There was times where it was thinnest and times where it was thickest. We also found that, you could look at just the average thickness for any digit. We could put this down to specific digits or digits per foot. And we could see, did any of these give us kind of unique results per foot or per digit and then per foot? And we also said, okay, we have so much data. Let's not worry about all the different, fine aspects. Let's just put it all together and say, is there kind of an average? So that was kind of the end thing that we did. And in the end, here's some of our, kind of interesting things, is that regardless of how we figured, all these different GWAS, we found basically the same QTL was identified for the front medial digit, which is on the front foot. So it didn't matter if we just put it into a front foot category, or we could look at a specific digit. Same QTL. Importantly, this is the digit that is the thickest, okay? Another thing that came out is we could also see variation here when we looked at average across time points. So particularly reflecting what time point, the sample time point 1 was when the digits are the thickest, sample time point 2 was when they were thinnest. So if we look at genetic regulation affecting digital cushions, it's a little bit different when we look at when animals have the thinnest digital cushion versus when animals have the thickest, I said that backwards, the thickest digital cushion versus the thinnest digital cushion. So again, showing us the complexity of biological regulation. In the end, we brought this back and we said, well, what genes are in these areas? And these are kind of, our results as far as SNPs, but then out of these results, there are 10 candidate genes that we found in these. And out of those candidate genes, based on annotation, we found 2 that seemed kind of biologically plausible in Holstein. And one of these is related to fat deposition. Another one is related to bone growth, which also has to do with the foot conformation, the hoof conformation. So that's kind of where we ended this part of the study in Holstein cows. But then we expanded the study and we said, well, how does this, change or vary if we compared Jersey cows? So we went back to that phenotype. And some of those, we said, what's the similarity and differences? So in general, Holstein had thicker digital cushions than Jersey cows. We also found an age correlation. So we could look at basically 3 years old was the key time point, that there was a significant difference, whether it was a cow or a bull, with digital cushion thickness once they hit the age of 3. Consistently, regardless of breed, regardless of sex, the hind medial digit was always the thinnest digit. So really important when we think about lameness. The front medial digit was generally the thickest in cows, but you could look at either front digit if you looked at the bulls. And I think it's a very important factor to think of just the difference in environmental factors impacting hoof trimming, locomotion, where the animals, how much they're moved, and housing between cows and bulls. We looked at variation by breed. So here's kind of some of our GWAS that we looked at in Jersey versus the GWAS that we did in cows. So this was kind of that previous study using that data, but it was comparing it over here to new data that we had received or gotten on Jersey animal. And you can see that there's some variation across breeds. So while, it could be something, again, that if you selected for this across breeds, you do have to take into account breed variation. In the end, when we compared Holstein and Jersey, we looked at, again, these regions and candidate genes. We found genes related to fat deposition, new genes, not just the ones that we had before, but new genes related to fat deposition, bone growth, as well as epidermal keratinocyte function. So that's kind of, putting all of this study together. And again, how we had, small sections of it, and then we built on that and expanded it. And there were 3 papers that came out from this particular research that gives you a lot more details on this one. Okay, the last cattle study I want to tell you about is our work in progress. So this is the main cattle study we have currently going on in my lab, and this particular study is being run by my graduate student, Azwan Jafar. So I have a passion for crossbred cattle, and in some countries this is very appealing. In other countries, this is definitely not a highlight for the country, including that in the US for many farmers. But I have a real interest in crossbred cattle, their performance, heterosis, and looking at how selection in crossbred cattle affects genome variation. So Azwan has a lot to do. With this in mind, our big picture objectives. So the first one is nothing novel, but it creates a foundation. So we are comparing pedigree breed estimations with genetic breed composition estimations. But we need this to start building on the next thing. So then we want to look at, again, how do these breed estimations of crossbred cattle, how does that relate to performance traits? And some of this has been done in different crossbred animals. But lastly, and I think this is real where the novel part of this project and the really exciting part is in our third objective. And this is the objective where we want to localize ancestry to actual chromosome regions and look at how this compares and correlates to performance and selection. So guys, the data I'm going to put out here on this one, this is preliminary data. We're still working on this. So just keep that in mind. As we go through these, I think important considerations, kind of going back to the title of this seminar, is I think it's really important for us to think about reference populations here, as well as breeding systems and the breeds that are used. So in my previous two things, we're talking about diseases and association studies and a lot about phenotypes. Now I'm going to be focusing more on what's unique about populations and why do we care, and how did some of these other things build into those populations? So this is a very collaborative study. We have 3 different crossbred data sets that are very different from one another. We're working with LIC in New Zealand, who have extremely well-established Kiwi cross in New Zealand, crosses of Holstein Friesian and Jersey. So, you know, I think the unique part here is that we have these animals that they breed kind of within this Kiwi cross group. They also use purebred animals still. We also have a population of US Pro Cross, as well as another group that's called Gray's Cross. I'm going to show you some data on. Now, this is a very different type of crossbred breeding system. It's a rotational crossbred breeding system. Very specifically, they use 3 breeds in this rotational crossbred system. So every generation, you use a purebred animal. So for instance, the Pro Cross is generally a cross of Holstein, Montbéliard, and Viking Red. So for instance, your very first generation will be a Holstein to a Montbéliard. That F1 produced from that will then be crossed into a purebred Viking Red. That next hybrid, the F2, will be crossed back to a purebred Holstein, and so on. The last group we're looking at are crossbred animals that happen to be sampled from organic herds across the US. So these are intentional crosses by the owners, but usually really just for an F1 generation, and then they're generally back crossing into the desired purebred breed, and really just looking for that hybrid vigor of the F1 generation. So we have 3 very different crossbreeding systems. So some of our preliminary data on this is, I think I wanted to put this up. I know it's not like super compelling because it's really just telling us what we know, but I think it's interesting because here we have preliminary data on the Kiwi cross, and it's looking at if we had our breed estimation and we used pedigree information. In all of the Kiwi cross that we have, you see kind of those Kiwi cross, some of them have, so down here is 0 amount Holstein, up here is 100% Holstein. And you have, you know, all this variation, and lots of them are around 50%, but you still have quite a bit of variation. And the mirror image of that is the percentage of Jersey composition in those same animals. Now compare this figure down here to this figure, and you see the genomic breed estimation of those same animals. And I think, you know, the big thing I want to point out is just to kind of show some of the difference here, you can see animals that from a genomic standpoint, we have animals that have basically almost no Holstein or Jersey to almost 100% Holstein and Jersey. And these are the same animals in these 2 analyses. This is jumping into the ProCross data set that we've done more work on. And one of the big things that we've assessed in this ProCross data set is, you know, when you do genomic breed estimations, so a lot of this work is using admixture software or some type of admixture software where we have our crossbred animals and then you have your purebred animals that you are kind of using as reference populations. And those reference populations, tell us then, or that's what we're using to figure out what is the breed composition of our crossbred animals. So in this figure, same crossbred animals, they're all pro-cross in this case. Well, there's pro-cross and there's graze cross. Graze cross are a similar 3-breed rotation, but they focus on Jersey, Viking Red, and Normandy. So focus more for a grazing animal. So over here, we have just out of all the animals we've sampled, which in these populations are usually almost 300 pro-cross and almost 200 of the graze cross. So over here's pedigree estimations. In this first set, well, that's actually in the middle, this is a genomic breed estimation, but it's using what we're calling the parental Viking Red as the reference population. This second set is another genomic breed estimation, but we use a different ancestral population to represent the Viking Red. So to add complexity to this breed estimation, Viking Red are a composite breed in and of themselves. So they are commercially marketed as Viking Red, but they are actually a composite of Finnish Ayrshire, Swedish Red, and Danish Red. So in these two systems, we use the exact same software, the exact same animals and thresholds. But in this particular one, to represent that Viking Red population reference ancestry group, we used animals that are commercially marketed as Viking Red, and these particular animals were the ones that were used as sires for our population. Whereas in the reference panel over here, we took out of publications European purebred animals that represented Finnish Ayrshire, Swedish Red, and Danish Red. So these are the breeds that were used to create and develop Viking Red, so very similar, but yet different. So the big thing here is when we use these two different reference populations, we want to point out that it significantly affected our breed estimations for the crossbred pro-cross and graze cross for every single breed other than the Jersey that were in the graze cross themselves. The Jersey were the only breed that was not affected, and it was specifically in the graze cross. So these different populations, reference populations, even though it was such a minor difference, had a significant effect on breed estimation. So that's really important because in the next step, we want to know how does breed estimations, how does breed composition relate to performance? So we had information on fat, protein, and milk yield, as well as somatic cell count in these animals. And we looked at breed composition estimates. Over here, we're using those ancestral ones of the Swedish, Danish, and Finnish animals. Over here, we have the parental commercial Viking Red. And the bottom line was, it actually came up a little bit different. So the one thing that was consistent is breed composition plays a significant role in production traits and somatic cell score. So for pro-cross, the amount of breed composition of the Holstein and the Montbéliard, where you see the asterisks, that was important for milk, fat, and protein in the pro-cross. And the breed composition amounts of the Normandy and the Viking Red was significantly associated with elite performance versus poor performance in somatic cell score. So breed composition played a significant role in relationship to traits of interest. So how does the reference population affect that association? And for us, I think the really unexpected result, even though it was significantly different, it did not have a significant difference related to performance. So it didn't matter which reference population we used in our data set, it did not affect how they were related to performance. So I think that was really important here. So it was different than what we expected. I think in different data sets, you might find that this still is significant as related to performance. So you have to think about the conclusions you draw based on the data sets you use. Okay, guys, I noticed this as I was going. I don't know what happened to my slide. Bear with me here. What this one is showing is that even though we have an extra slide here, is that this is kind of where we are now. So this is extremely preliminary. And what we're doing is we're we're localizing ancestry across all the chromosomes. So what these pictures are showing you is each horizontal line. So this is chromosome 1, chromosome 2, and it goes down through all the chromosomes. And the different colors, whether it's red, blue, green, or gray, represent different breeds, whether it's Holstein, Jersey, Montbéliard, Normandy, whatever's the most appropriate one there. Now, the key to what we're trying to find here is comparing our elite animals and our poor performing animals and saying in both categories, whether it's elite animals, we have elite animals that have 90% Holstein, we have elite animals that have 20% Holstein. We have poor performing animals of the same trait that have 80% Holstein and, you know, about 5% Holstein. So the amount of variation within a group is huge. Even though with that variation, we're still seeing significant association to ancestry. Now we want to pinpoint that and say on a chromosome level, can we really kind of pinpoint this and say, even in, you know, the high performing animals that only have 20% Holstein, is that Holstein ancestry over really important genes? And this is kind of where results are leading us in this idea. So our preliminary analysis, we're finding that we have blocks of Holstein that is over, you know, certain regions that are already known QTL for production traits. In some of our preliminary results for the Gray's cross, we're finding, again, these blocks of certain ancestries over known QTL for health traits. So it's leading us in this direction. I will say these particular results, while I'm super excited about them, we have a lot of work to do on there. One of our biggest challenges is we can do this localization to chromosome for individual animals. That works great. How we can make kind of a composite animal that represents a group, such as the elite or the poor performing animals, that's where we're struggling. Or how do we put all of our individual results together? That's where we're struggling. So this is a work in progress, and I'm saddened that I can't tell you or show you more because I think it's the most exciting part, but we're getting there. So in all, I think, you know, the big things here on the crossbred stuff is results reflect variation that we expect between genetic or pedigree and genomic stuff, but reference populations can really play a role in this. We're seeing that ancestry is related to traits of interest, and preliminary results are, you know, compelling and exciting related to ancestry and chromosomal regions as well as selection. So that's kind of where we are there. I'm gonna skip a couple of slides and go to the very end. I do work on dogs, but you know, you'll have to come back or invite me back for a different one on dogs. And I just want to say that of all of these, I think, you know, other than just the research results we had, my biggest things are think about how we capture and analyze our data, both on the phenotypic front as well as the genetic analysis. Think about the impact of populations in our approach and how this relates to biology and selection. Sometimes we really get stuck in just being computer geneticist. And also think about the application across species and disciplines. And that's where, as silly as it sounds, I work on dairy cattle and I work on working dogs. And there's a lot of things that are complementary. So I took up lots of your time. I am happy to stay longer and answer questions. And that's all I have, guys.
All right. Thank you so much, Dr. Huson. It was a great presentation. And now you in the audience, if you have a question, you may unmute yourself and raise your hand so I can see you here and call your name. I have already some hands up. So yeah, Marie, if you want to go, please.
This is just tremendous. I was very impressed with all the information and work you're doing. And my question isn't about the results. It's just you went very fast and I wasn't able to keep up to all that. And so we'll be able to, for people that go at a slower pace than you do, be able to kind of review it. Thank you very much. You did a tremendous job. Thank you.
Thank you so much. And I'm happy to share my slides or the references and stuff here. So you can look into it further, and you can always email me.
Thank you. Thank you very much.
Right, next one, I see Flavio, if you want to go.
Okay. Can you hear me?
Yes.
Thank you so much for this presentation. Excellent. And I have one question about your third study on the crossbreds.
Mhm.
I was surprised, Heather, that you didn't account for or try to estimate the epistatic loss in those crosses.
I'm sorry, could you repeat that? I didn't quite capture it.
Epistatic loss? Because you are working with the crossbreds, right? Rotational or composite breeds. So you are going to have a loss due to the recombination, or you're going to lose epistatic effects. So you can, if you know the breed composition, you can easily calculate the expected epistatic loss, because you have like heterosis, right?
Yep.
But you are going to lose, especially in this from the hosting side. You're going to lose a lot of ability of those animals to produce milk, for instance, because of the epistatic effects that were fixed by selection in the hosting breed are going to be lost with time as you cross, right?
Yes.
So, and
yes.
then when you show the results with the Manhattan plots there, so it's not surprising to me that you have a variation of animals, the top animals with 80% hosting and some 5-10. That could be due to the epistatic loss for the individuals, right? So, and because you have these different compositions, you could estimate that in putting account in your model.
I think I need to catch up with you after this and talk through that, because we're struggling, especially with where we're at localizing it per chromosome, and thinking about, recombination and, some of those really important like haplotypes that are fixed in purebreds that are being broken up in the crossbreeds. And we're struggling with some of that on the chromosomal part in particular. So I'll be emailing.
Yeah, I think you could do something very quantitative first, just to include in the model. Probably you are including heterosis already, right?
Mm-hmm.
But include the coefficients for the epistatic loss. And then you're going to have idea if this is could play an important role, right?
Yep.
To trying to understand the genomic results better. Just a suggestion, because if you look work done 50 years ago, 40 years ago in crossbreeding, there are a lot of studies in epistatic loss, right?
Mm-hmm.
When especially when you cross these breeds that are very productive, like hosting.
Right.
And maybe, I'm not saying that jersey is not productive, but I mean, they have very different levels of production, right? And they have these specific effects that were selected for many years within each breed. And then you cross them and you keep crossing, you keep losing those
Exactly.
recombination, right?
Yep. And I think the different types of crossbreeding, whether they're using like that rotational crossbreeding and hence a purebred every year versus kind of more of creating the composite, I think that's really interesting. And I know I want to do more there. We haven't gotten to that point yet, but I really want to see the effects of the different styles or the systems on the genome.
Yeah, and with the Kiwi data set is different because they are selecting the crossbred animal, right? So they're kind of recovering those loss. But in the case of the rotational cross, there is no selection, right? They are not using the crossbred. They are not selecting.
Not heavily. Not heavily.
So they are not trying. They are not recovering that loss of due to the side effect.
Mm-hmm.
Yeah. So just as you guessed, great presentation, great
Thank
work. Congratulations, Heather.
Thank you, Flavio. And now I see a last hand, Gerson, if you want to go.
Hello, Dr. Heather. Thank you very much for the presentation. It was amazing, the amount of work. I have two really basic and easy questions for you.
Okay.
And I will start with the third chapter, let's say, the third presentation. That I was just curious that I worked a little bit with this admixture analysis. And I was thinking how, because you have some really similar breeds, like the reds, and I suppose that they share a lot of haplotypes. And I was wondering how precise you can separate these, and then in your cross animals say like, no, this is Danish red and this is, I don't know, because I guess that they share lots of haplotypes. And anyway, this is something that I was questioning myself here.
For our analysis and in our purpose, we were just trying to characterize kind of that Viking red ancestry. So in this particular analysis, we had it easy where we didn't have to separate what was Danish, Finnish, or whatever the other one was.
Norway again? No.
Yeah. So we didn't have to separate them because all of them were contributing to that Viking red ancestry. So we actually added them. I will say when we did do the admixture analysis, the Danish red were more distinct and kind of pulled out their own genetic signature as compared to the Finnish Ayrshire and the Norwegian red. Those two were almost identical, and we couldn't separate them in our analysis. It actually started pulling out little patterns in other breeds instead of separating those two. But in our results, we didn't have to separate them because they all contributed to Viking red.
Okay. And I know that we are over time here, but this is also a quick and easy. On the soul fat thickness, I forgot the proper name, I'm sorry. And I'm sorry if I missed this, but I'm just curious if you have the correlation of that trait with the hoof health trait that we have here in Canada as an example, if they have like a good correlation or maybe you mentioned this, but I don't know if I missed it.
Which trait were you asking about?
The
digital.
Yes, exactly.
Cushion thickness. I don't know that you guys have that trait, and the US doesn't technically have that trait. Now they select for like foot and leg composition. Some programs do a lameness score, but we don't have that trait currently in any of our genetic selection programs. So it's a complete new novel trait that if the industry would choose to do it, we could do it. Limitations are, we used ultrasound. While we narrowed it down and we said, well, here is the digit on the foot and at what time you should do it, it still would be a labor-intensive type of thing to actually score on lots of animals. So that's the limitation for potentially incorporating that down the road.
Yeah. All right. Thank you. Bye.
Yep.
All right, Filippo. And then we conclude our meeting. Yes,
thank you so much, Heather. Gotta say, just a fantastic presentation. You are always so clear. Always love you. You design your experiment, makes sense, and you tell such a nice story. So I really appreciate. Gerson still part of my question. At least, can you tell us what was the correlation, the magnitude of correlation between BCS and DCT?
So it was tough in our original, the phase 1, where we only had like 170 animals. There was no correlation in that data set. And now the previous work done by Rodrigo Bacallo, he found an initial correlation. That initial phase of ours, we didn't. When we increased our data set, up to, I think we were 500, 600 animals for that data set, and we were cross farms, then we did have a correlation, and it was, as you would expect, lower body composition or lower body condition, thinner digital cushion. So, I mean, big picture, you feel like the animal just in general has less fat reserves.
Anything with heel depth or foot angle, those are the traits that I guess Austin USA would
We didn't do those traits on these animals. And I think the biggest challenge we ran into, because we were really interested on just leg conformation when we started that study. And the bottom line was, unless you'd set it up in a much better research, very strict way you do it, there was too much variation across farm, how they hoof trim, when they hoof trim, how often they hoof trim. Yes, we just didn't do that one.
Thank you so much. Hope to see you in summer meetings. Anyway, thank you again.
I hope so.
All right. So once again, thank you so much, Dr. Huson, for accepting our invitation. It was a great presentation. And yeah, I hope to see you all next week as we continue our series of seminars. Thank you so much. Have a great afternoon.
You are welcome to give out my email if you want, if anyone has follow-up questions. I know that was long and a lot of info, so people are welcome to shoot me an email.
Awesome. And I will make available the records if you want to hear again or see again. Just shoot me an email for the CGIL email, and we can provide that for you. Thank you so much.
Thank you.